Getting science wrong is just part of the scientific endeavor. We're never going to perfectly understand the world. We just keep trying, and hopefully, over time, we develop a closer and closer approximation of the world as it is. But can science be so wrong that it should be taken back? That's the hot controversy rocking the microbiology world right now, as the prestigious journal Science retracts a 15-year-old paper that thought it discovered arsenic-based life. Spoilers. It did not. First, let's talk about the original 2010 paper. Scientists were studying a strain of bacteria isolated from Mono Lake, California. Mono Lake is alkaline and rich in arsenic. For most species, including humans, arsenic is toxic as hell. But in Mono Lake, once again, life finds a way. There are species of nematodes with extreme arsenic resistance, tolerating concentrations 500 times what would kill a human. Others chemically convert it for their own use. None of that is super surprising. There's selective pressure to try to make all sorts of environmental circumstances work, and this is just one more. But according to these researchers, bacterial species GFAJ1 could do something completely novel: incorporate arsenic into their DNA. The idea is that life needs things to live, and life on Earth needs 6 things in particular: carbon, hydrogen, nitrogen, oxygen, sulfur, and phosphorus. Individual species may need more than that, like we use zinc and iron and copper and all that, but if you are life, you need those things at minimum. DNA alone uses 5 of them: hydrogen, oxygen, nitrogen, carbon, and phosphorus. Phosphorus is part of the sugar-phosphate backbone of DNA, so it's pretty mission-critical to the structure of the whole thing. And even though phosphorus availability can sometimes be a rate-limiting factor in life growth, you'll note the phosphate in some types of fertilizers, phosphorus is a comparatively abundant element on Earth. In the periodic table, phosphorus is in the same column as nitrogen, and, you might notice, immediately above arsenic. It's not a hard and fast thing, but a lot of the time, elements in the same column of the periodic table have similar properties, including for biochemical purposes. For instance, bromine is a halogen, like iodine below it, and its properties are similar enough that our thyroid, which uses iodine, can take up bromine instead. Of course, that goes poorly, because our bodies might not know it, but they don't actually need or want bromine. It's just different enough from iodine to be a problem. It's a similar sort of idea here. In fact, one of the things that makes arsenic such a toxic problem is that phosphate and arsenate are just similar enough to many of our metabolic pathways to be dangerous. But plenty of species can adapt to all sorts of chemical circumstances. After all, this bacterium, GFAJ1, was in an arsenic-rich environment, and arsenic is similar enough chemically that researchers thought it could deal with the chemical dissimilarities and successfully incorporate arsenate in place of its more stable phosphate-based counterpart. To test their hypothesis, they effectively grew these microbes in a medium where phosphate was ultimately made unavailable, but arsenate was still there. Using radio-labeled arsenate, the radio label lets them know for sure that it's what they added, they gave it to these bacteria, then broke them up into different fractions of biomolecules, proteins, lipids, nucleic acids, and so on, to see where the radio-labeled arsenate had gone. They reported that their radio-labeled arsenate was incorporated into the purified DNA fraction. These bacteria didn't require arsenic. They reportedly grew far better with phosphate, but it seemed like they could hot swap one for the other as a quirk of evolving to thrive in their arsenic-rich home. And if you read the paper, you can absolutely see how it could have cleared peer review and be published. It's a wacky hypothesis, but it seems plausible on the surface, and they do a bunch of experiments to try to support the idea. It's exactly the kind of out-there science that, when true, shoots up to the top journals for publication. And so it did in Science in December 2010. The problem is, and this is true, arsenic is not phosphorus. A sugar arsenate backbone is intrinsically less chemically stable than a sugar phosphate one. And while these were good researchers, first author Felisa Simons, a NASA astrobiology research fellow at the U.S. Geological Survey at the time, they were nonetheless in a real extraordinary claims require extraordinary evidence kind of situation. In this case, that means a solid claim of arsenic-based life requires ruling out other, more plausible explanations. For instance, that the bacteria were tolerating the arsenic and hanging on to it, but not necessarily substituting the phosphate. The author's own data actually kind of supported this idea. One electron microscopy picture shows the cells have these vacuole structures, which can often be a sign that the cell is trying to sequester toxic material. Another potential confounding factor was studying the cells when they were in resting phase rather than active growth. Growth involves lots of RNA production, and RNA, like DNA, requires a lot of phosphate. So if you're studying these cells when they don't need hella phosphate, how can you be sure your cells can actually hot swap the arsenate successfully? These criticisms are characteristic of the response to the paper at the time. No one's saying their data are faked. They're just interpreting them incorrectly. That makes sense because what's more likely? That a bacterium can thrive with far less chemically stable DNA bonds that are way more likely to break apart in water, or that a bacterium is highly arsenic resistant but can't actually use arsenate in place of phosphate? Eventually, pretty much everyone in microbiology came to the conclusion that it was the latter, particularly when follow-up studies demonstrated that GFAJ1 can survive with a lot of arsenic and not a lot of phosphate. But no, arsenate wasn't in its DNA. And this paper's particular flavor of wrong isn't one we have a fantastic way of handling in science. As the recent retraction notice confirms, retractions have historically been for fraudulent papers or scientific misconduct. There are no shortage of retraction scandals like those, and they can hit any corner of science, from Alzheimer's research and amyloid beta star 56 to physics and room temperature superconductors. Retractions are a big deal for exactly this reason. They typically indicate that someone did a bad and intentionally published misleading, wrong science. There's absolutely no indication that this was the case here, and so far as I can see, no one's alleging that. On the other side of the spectrum, a publisher may issue corrections. These don't carry the stigma of retraction at all. Usually they're for little oopsies. We meant to refer to figure 4, and we accidentally called it figure 3. It's the kind of thing that gets a correction. Sometimes they're a little bigger than that, and can even be a little egg on the face, like, we meant to submit figure 4, but we actually published figure 3 twice. Where you really would have hoped someone noticed before publication, but the presumption is that a correction is not intentionally misleading. It's usually a little thing, and it's typically about the presentation of the manuscript, not something wrong in the data analysis or interpretation themselves. And in between these, you have a whole range of being wrong. There's a glorious, rich tradition of scientific debate that goes something along the lines of, beautiful findings you've got there. You're interpreting them wrong, though. Let me do a few more experiments to show you why your data actually prove a completely different point than you thought it did. Sometimes these back and forth exchanges are scientific subtweeting. Sometimes researchers will straight up flex on their colleagues. But the assumption is that all parties are in it to find the truth. We're all here in good faith. All these observations are true. We just contextualize them and understand them differently. The arsenic-based life paper pretty cleanly falls into that last category. Yes, the bacteria took up the arsenate, and yes, further experiments would show that it wasn't actually incorporated into the DNA or RNA. It is neither fraud nor typo, neither a traditional retraction nor a traditional correction. So scientists hashed it out in the comments section, basically. There's only one problem. When a paper is retracted or corrected, there's always a notice. Retracted papers have a big, all caps, "retracted" on them. And most digital platforms will put a correction notice right at the top of the page, even before the paper's abstract. But until now, if you looked up the 2010 arsenic-based Life paper, from the paper alone, you'd have no idea that the overwhelming consensus in the field is that this paper is wildly, wildly wrong. You'd have to find and read all the follow-ups to know that's the case. So basically, a paper like this becomes a newbie trap. If you don't already know these findings have been debunked, you could easily read it and go, oh, cool, arsenic-based life is a thing. It ends up perpetuating the mistake. Even though this is way more than a, whoops, we meant figure 3, there's actually less notice about its wrongness than with that kind of mistake. Arsenic-based life falls into an uncanny valley of wrongness. Ultimately, the editors of Science say that standards have changed for retraction. Even without data manipulation, this is a case where, quote, the paper's reported experiments do not support its key conclusions, even if no fraud or manipulation occurred. By officially retracting the paper, Science's editors are trying to prevent the newbie trap and give readers some kind of warning not to believe the paper's findings. At the same time, I understand why the original study's authors oppose this retraction. It stigmatizes a finding that was published in good faith, and it lumps these authors in with the frauds and cheats. Being wrong isn't a science crime. It's part of the process, and that shouldn't be punished. Reinterpreting true observations is one of the most beautiful things in the scientific endeavor. As the study's authors say in their response, quote, claims should be made, tested, challenged, and ultimately judged on the scientific merits by the scientific community itself. So how do we deal with these problems? The preponderance of the evidence absolutely demonstrates that no, GFAJ1 is not arsenic-based life. But even with incredibly broad scientific consensus, even if 99% of scientists agree, there's still no one true arbiter of science who can say, this paper is wrong. But as we keep years of papers in easily accessible digital archives, we're gonna need more tools to avoid newbie traps, to signal to readers, hey, you should know before you read this that scientists in this field, or even the authors themselves, now disagree. Something that isn't a retraction, isn't a correction, but still informs readers as to the current state of the game. We don't have that now, and as a result, arsenic-based life has been retracted. Even if maybe it shouldn't have been retracted about it. There are lots of ways to be wrong, after all. I should know.